Hello, everyone. We have a new session of Sybeco. Our today's guest is Giulio Bondanelli. Giulio did his bachelor's in physics at the University of Bologna in Italy, and then an international master in physics of complex systems between Turin and Paris. He then switched to computational neuroscience for his PhD in Paris at ENS with Serdijan Ostojic. And now he's doing his postdoc with Stefano Panzeri at University Medical Center Eppendorf in Hamburg. He's interested in knowing how computational, complex computations and behaviors emerge from the structure of neural circuits and population dynamics. And to address this question, he tightly collaborates with experimental labs to develop data-driven models of neural circuits. Thank you so much, Giulio, for accepting our invitation.
Thank you very much.
Sure.
Yeah, thank you for the nice introduction and for the invitation. So I'm gonna share my screen. 1 sec. Okay, can you see it?
Yes, it's all good.
All right. Okay, today I'm gonna talk to you about this project about synaptic motif in the association cortex that support decision making. So I'm mostly interested in how brain circuits, mostly in cortical areas, generate specific kind of neurodynamics that then support specific computation or specific behavior. So the overarching question that I'm interested in is how the computation arises from the local network connectivity. And so this computation in general likely reflect functional differences across brain regions. So you might think that sensory areas, for example, if you think about V1, might do something like coding of contrast or orientation coding. So it might privilege some kind of faithful representation of external stimuli. But then these external stimuli must be integrated and represented in an abstract way. So there are other areas, association areas, that might devoted to do this kind of stimulus integration and abstract representation of stimuli. And then maybe motor areas generate motor preparation or pattern of activity that can support the generation of specific movement or action selection. And so people in computational neuroscience have explored different kind of models that implement these different computations, and all these models have different structural features, meaning different properties of local connectivity that support this computation. For example, in sensory areas, mostly V1, people have looked at ring model for coding of orientation in association areas. People have looked at these models based on mutual inhibition between selective populations. So this is a seminal work from Xiaojing Wang, where you have different population coding for different choices that are mutually inhibited so that they can support stimulus integration and working memory at the same time. Or for motor areas, people have looked at these inhibition stabilized networks which have strong excitatory connectivity, which is then stabilized by feedback inhibition. And these are models that generate strongly transient activity that can support different motor actions. And so the question that we are interested in is how is connectivity organized in different local circuits, and whether we can measure this structure in experiments, and how we can make sense of it. And so people have mostly looked at this in visual areas, mostly in V1, visual cortex, and mostly for excitatory connectivity. So these are two works that have tried to correlate the connectivity strength of visual cortical neurons with functional properties of these neurons. So in this paper, they computed the connection strength between pairs of neurons as a function of the response correlation, which is the correlation between the neural activity of pairs of neurons. And what they saw is that excitatory neurons which has similar response properties are more strongly connected. And in another work have shown that neurons that prefer similar orientation were more likely to form synapses. So excitatory neurons with similar selectivity are more frequently connected. And so what these work suggests is that you have this like-to-like excitatory to excitatory motif where excitatory neurons connect preferentially with other excitatory neurons that share similar functional properties. But so people have also gone beyond only excitatory neurons, and they've looked at both excitatory and inhibitory connectivity. So this is recent work which has measured the EPSP and IPSP from a pair of excitatory and inhibitory neurons. And what they found is that the excitatory postsynaptic potential strongly correlated with the inhibitory postsynaptic potential. And this in pairs of excitatory and inhibitory neurons. And this means that excitatory neurons connect preferentially with inhibitory neurons from which they receive the strongest feedback inhibition. So this forms kind of co-tune or co-selective or like-to-like ensembles of excitatory and inhibitory pairs of neurons. And so we have some evidence of how the connectivity is organized in cortical circuits, mostly coming from anatomy or electrophysiology. So the pattern that starts to emerge is that in early sensory areas, mostly V1, we have this co-selective or co-selective ENI connectivity that may support this sharpening of sensory responses in early sensory areas. But we don't really know what is the connectivity, how is the connectivity structure in association areas closer to the behavioral output. And so this is the main question that I will address here. So the question is, what is the organization of excitatory and inhibitory connectivity in association areas? And so I just want to thank, I had my collaborators, Kuan, who collected the data, and Laura Driscoll, who collected another set of data previously from the same brain that I'm going to show. Dan Wilson, who collected data about an experiment that I will show you at the end, and other people that contributed to this project, and the PI, Stefano, Chris, and Wei. So this work is based on a multi-level approach for understanding decision-related computation. And so what we did is to combine a behavioral task. So we have behavioral data. We have two-photon population recordings from association areas, and then we have data from connectomics using electron microscopy and network modeling to interpret this set of data. So the task that we looked at is a perceptual decision-making task. So this is a navigation-based decision-making task where we have a T maze, and we have a cue at the beginning of the maze, a visual cue that the mice see, and then we have a delay period during which the animal have to remember the visual cue, and then we have the turn where the mouse has to turn left or right depending on the visual cue. And so we looked at an area which is the posterior parietal cortex. This area has been shown to be necessary during this kind of navigation-based task. And the typical neural activity that you see during this kind of task is the following. So the previous work from the Harvey lab has characterized in depth this the neural activity during this kind of task. And what you see during this task is that you have neurons that are selective for different cues, so different stimuli or different choices. And not only that, but selective neurons are also active in specific epochs of the task. So if you plot neural activity across the trial duration, you see that this kind of choice selective sequences that tile the whole stimulus duration. So there are these two things. So one is choice specificity of neural responses, and one is this temporal organization in neural sequences. So what we did is the following. So they preserved the brain from an animal that was previously trained in this kind of task, and then we did my collaborators did electron microscopy on this local piece of PPC circuit. So they used electron microscopy to map anatomically all the neurons and connection within this volume. And what we have is a set of about 130 neurons, excitatory, inhibitory, and around 200 synapses, both from excitatory to inhibitory and inhibitory to excitatory. And so we have connectomic data, but also, as I said, two photon population calcium imaging data from the same task. So we managed to map the anatomically traced neurons with the imaged neurons. And again, we have this is just an example of the neural activity that you have in this kind of task. So it's trial type or choice selective, and it is temporally selective as well. And just interrupt me at any time if you have any question. I forgot to say.
Giulio, the inhibitory neurons are just broad inhibitory. You guys did not specify the cell types.
No, we didn't specify further. At some point, we broke it down into basket and Marinotti cells, but given that we have not so much data doing statistics, when you break down things, it becomes a bit difficult. So,
thanks.
And so, given the connectomics data, we quantified the pairwise anatomical local connectivity between pairs of neurons. And the way we did that is by looking at this normalized synapse frequency, which is given a pair of neurons, is defined as the number of synapses between this pair of neurons divided by the overlap between the axon of the presynaptic neurons and the dendrite of the postsynaptic neurons. So it's kind of a measure of the connection affinity between two pairs of cells, or the probability that they make a synapse, because you normalize by this cable overlap. And on the right, you just have an example of a pair of neurons with a dendrite, axon, and synapse in blue. And so what we found is in PPC, by looking at the selectivity of excitatory inhibitory neuron, we found that they're equally selective, and I think that's also what you find in your paper. We didn't find any significant difference between the selectivity of excitatory and inhibitory neuron, meaning that also inhibitory neuron were selective for the choice or the trial type or stimulus group. And so what we did to measure the functional similarity between any pair of neurons is to consider the mutual information between the neural response of a given neuron and the stimulus. So you would do that for example, neuron I and neuron J. So you have it's just two measures of selectivity, and then you combine those measures as it's just a geometric mean or the product between these two selectivity to give you a measure of how similarly tuned are those a given pair of neurons. So if neuron I and neuron J are selected for the same stimulus, this selectivity similarity would be positive. If they're tuned to opposite stimuli, this measure would be negative. And if one of them is non-selective, this measure would be close to 0. Okay, so what we found is that, so we looked separately at excitatory to inhibitory connectivity and inhibitory to excitatory connectivity. And what we found is that when you break down pairs that are pairs of neurons that are selective for the same stimulus, so this would be the co-neuron pairs, and neurons that are selective for opposite stimuli, that would be the anti-neuron pairs, you found that the normalized synapse frequency is larger for the co-tuned pairs of neurons than the anti-pair of neurons. And if you look at another measure of connection strand, the PST area, you also find the same result. So this is just by binarizing this measure of functional similarity of a given pair of neurons, but you can do it in a continuous way by looking at the connection strength as a function of this graded measure of functional selectivity similarity. So what we found in this case is that the E to I connectivity strength is positively correlated with the selectivity similarity for pairs of neurons.
Giulio, question.
Yeah.
So this is more, I'm trying to understand the experimental side here. So 2-photon experiments are done, and we know excitatory and inhibitory from those experiments.
We know from the morphology, actually.
Okay, so then
you don't know from the...
I see. So then there is some microscopy. I see. Then there is this in vitro microscopy, and from the morphology, they tell apart excitatory, inhibitory, and then EM follows that. So these are based on EM results, or is this? Yeah,
so you do the EM, and then from the morphology given by the EM, you find excitatory, inhibitory. Yes. Yeah.
Okay, I see. So it's not like you guys register between the 2-photon and the EM.
No, so they were done in separate phases, and then when we put together this data, we co-registered EM and 2-photon, but not for the ENI. So the ENI were just derived from the morphology.
I see. And the morphology comes from the electron microscopy.
Yeah.
Okay. Thanks.
Yep. Okay, so what we found is that excitatory to inhibitory connectivity is co-selective, meaning that excitatory neuron projects more strongly to inhibitory neuron that prefer the same stimulus, or were more functionally similar, tuned to the same stimulus. So then you can do the same for inhibitory to excitatory connectivity, and what you find is the opposite. So the connection strength is weaker for co-selective pair and stronger for anti-selective pair. And if you look at this graded selectivity similarity, you find negative correlation between connectivity strength and function similarity, meaning that you have anti-selective I2E. Sorry, this is a typo, I2E. So what we have is co-selective excitatory to inhibitory and anti-selective inhibitory to excitatory connectivity. And so to kind of put this together, we looked at a excitatory inhibitory recurrent network models. And so to start with, we looked at the linear recurrent network models that had two subpopulation of excitatory and inhibitory neurons, or population where you could tune the different the selectivity of I2E or E2I. And so what you find in this in these models is that if you have opponent inhibition given by co-selective E2I and anti-selective I2E, as in the data, what happens is that if you give input on, let's say, the left population, then you excite the left selective population, and then through this opponent inhibition motif, you inhibit the right selective population, and then in turn, because of this mutual inhibition, you amplify the activity of the left selective population. So you can work out the mathematics of this model analytically, and what you find is that you can write the response of the network models, and without going into details, you find that the network response is a function of both the mean connection strength of each connection type, which is these terms S, which is the sum of the connectivity that is within the same population and the connectivity that goes across choice-selective population, and the selectivity of this connection, which is this term delta, which is the difference between the strength of within pool connectivity and across pool connectivity. So basically those two terms, so the mean connection strength and the connection selectivity, give you the response of the network to external inputs. And so what you can do is to write basically the distance in the state space. So imagine you have these two dimensions which correspond to the excitatory population selective for right stimulus and the excitatory population selective for the left stimulus. Then what you can write is the distance in the state space of the output response, so the response of the network to the external input, and the distance between choice selective responses just due to the external input. And this gives you, this value is basically how much the recurrent connectivity enhanced the separation between trial type specific mean responses in the network. So how much the recurrent connectivity amplifies the separation between trial type in the neural state space. And so what you find is that this quantity here just depends on the selectivity, and in particular on the product between the selectivity of E2I and the selectivity of I2E connectivity. And what you can do is to try to decode the trial type as a function of these two quantities, so the E2I selectivity and I2E selectivity. And then what you find is that in the opponent inhibition regime that we identified in the data, which corresponds to co-selective E2I and anti-selective I2E connectivity, you have amplification of the choice-specific mean responses, meaning that in the opponent inhibition regime, what the network does is separating those choice-specific responses, and this may allow for a higher decoding accuracy in presence of noise. So so network with opponent inhibition increases the separation between trial types in the neural state space, which allows amplification and better decoding accuracy.
Giulio, sorry, could
Yes.
you go back? I want to make sure I understand this figure. So you're saying that we get unstable network if I2E selectivity is too negative and E2I is...
Yeah, because at some point, so if you make the I2E selectivity too negative, then your opponent inhibition motif is too strong. And given that it's a linear network, the mutual inhibition, which acts like effectively as a positive feedback, because it amplifies the signal, will make the system unstable because the mutual inhibition.
Right, but okay, so fair enough. But should we not get unstable regime when we have two positive I2E and two positive E2I? Because it's like if they're both positive, so how come we don't get that?
I mean, so you see that basically you have this unstable. I mean, if you fix, for example, if you fix the I2E selectivity here, then you also get an instability if you increase the E2I selectivity. So both.
Yeah, but then what is blue here?
Here?
Yes.
So this blue is the regime where the I2E selectivity is co-selective, meaning that you have so you have both E2I and I2E co-selective. Yes. So that would basically shrink the trial type's specific activity in the neural state space. So it's the opposite of amplification. So in the point inhibition, you have amplification. So they are pushed farther apart. In this regime here, they are shrinked basically in the state space because you have feedback inhibition in the same pool. So it would be this case here.
The positive I2E selectivity would mean that it's like-to-like inhib.
Yes.
Right.
Yeah.
Right. And then you're saying the decoder accuracy is lower there because we kind of don't have the competition between the two populations.
Exactly. Yes. Yeah. That's correct.
Okay. I just would have expected the network to be also unstable if we have two positive I2E. So basically, I would have expected that unstable to also exist on the top right corner.
No, because so the instability really arises from this this mutual inhibition. So if you have like, in this extreme here where you have co-selective I2E,
because they inhibit each other.
Then it's just a feedback inhibition.
Yes,
so
I see your point. Yeah. Okay, thank you.
Yeah, sure. Okay, so this was just a very simple linear rate model. Then we can go further and ask whether more biologically plausible network models that resemble PPC activity can also support these conclusions. So what we did is to fit excitatory inhibitory recurrent network models to the PPC data. And so where the identity and number of excitatory inhibitor neuron was matched to the data. So we fitted these network models to the neural responses of the coming from the two-photon PPC neural activity. And so here is just an example of the two-photon data in color for one excitatory, one inhibitory neurons and the fit on the top. And if you look at the connectivity of the fitted networks, you find that naturally you find co-selective E2I and anti-selective I2E as in the data across all network realizations. So the fitted network naturally exhibit this co-selective E2I and anti-selective I2E that we also see in the data. And so one prediction of the linear rate model is that if you increase the E2I selectivity, you get amplification of the trial type specific responses and so enhanced decoding accuracy. And the same if you decrease the I2E selectivity. So if you make them more anti-selective, you have this simplification of responses. So we tried to test whether this prediction was held in the fitted RNN. So what we did is to take the connectivity matrix that came out from the fit to the PPC data, and then perturb it a little bit in a way that you could increase or decrease the selectivity of different connectivity. So in the first panel, we perturb the E2I selectivity, and in the second panel, we perturb the I2E selectivity. And so as predicted by the linear rate model, increase in the E2I selectivity amplify the distance between the trial type specific neural activity, and the same if you decrease the I2E selectivity. So this showed that this opponent inhibition motif that is present in the fitted RNN also had similar effect, or may support the encoding of choice through this mutual inhibition motif.
Question: the RNN was fed to data at what time point? At all time points. So you have the
I see.
And then course of. So this E2I and I2E connectivity was something that emerged from the network. You guys didn't.
So we matched the identity of the ENI neurons on the model. So the model had pre-specified inhibitory and excitatory neurons that matched in number.
Numbers, yes, I saw that note, but I'm saying in terms of connectivity. So you enforce this. The connectivity was something that emerged in the network?
No, so we enforced ENI connectivity. So Dale's law was enforced in the network. What emerged is the co-selectivity and anti-selectivity of E2I and I2E.
Okay.
But the Dale's law was enforced in the network before it. Yeah. All right. So this kind of suggests this opponent inhibition that we see in the data could be used by the network to support some robust encoding of the choice in PPC. So one, as I said at the beginning, so there are two features of neural activity in PPC. One is the selectivity for the choice of neural dynamics, and the other one is the temporal organization of neural responses, meaning these choice selective sequences. So what we find in the connectomics data actually is that if you consider a pair of neurons, they could be active at the same time. So this, for example, this pair of neurons, INJ, is selective for choice 1 because they have higher activity for choice 1, but also it's their neural activity has a peak that is that happens at the same time, right? And this example here is still a pair of neurons that is selective for choice 1, but the activity peak happen at different times. So there are pairs of neurons that are active at the same time and pairs of neurons that are active different times. And so what we found is in the connectomics data is that for both ENI connectivity, for both ENI and I2E connectivity, we found that the majority of connection came from neurons that are active at different times. And so we wonder what is the role of this connectivity between neuron active at different times, since you might, for example, think that the network wants to have more connection between neurons that are active at the same time. And so what, to try to make sense of this, we looked at the fitted RNN models, and we computed the currents to postsynaptic neurons, and coming from excitatory and inhibitory neurons, active either at the same time or at different times. And then align the current to the peak of the postsynaptic neurons. And so what the postsynaptic neuron sees is that it is a strong current coming from inhibitory neurons that are active at different times. And so if you perturb this connectivity between neurons active at different times, so what we did is a manipulation on the fitted connectivity and then regenerate the neural activity in the fitted networks. And so if you reduce the strength of same connection and measure the sequentiality of the network, you find no change with respect to the fitted networks. But if you reduce the strength of the connectivity between neurons active at different times, you have a large drop in the sequentiality of the regenerated neural activity. So this basically means that, or suggests that connection between neurons that are active at different epochs within the task has possibly a role in generating sequences of neural activity. So what this means is basically to keep the postsynaptic neuron shut or low activity after their peak, you need this strong inhibitory connection, and these connections are from inhibitory neurons that are active at different time with respect to the postsynaptic neuron. And so you can do other manipulation in the fitted network. You can reduce the, this is just an example. So if you reduce same connection and you regenerate activity, you see similar activity. But if you reduce stiff connections, the sequentiality is degraded. And also if you fit network model without connectivity between neurons active at different times, the network cannot fit anymore. So meaning that these connections that are prominent in number in the data set, so between neurons that are active at different time, may support these child-specific sequences that we see in the posterior parietal cortex during the task.
Question here, Giulio. So, okay, if I understood, you're trying to come up with a mechanism for how we get these neurons tiled in the whole child window. And then, yeah, I'm not sure. I don't really understand this figure on the left side, whether for excitatory or inhibitory, but you're saying that we have an excitatory neuron and it receives input from multiple inhibitory neurons.
Mm-hmm.
And then you're saying that these inhibitory neurons being active at different time points, that's how we get this sequence, or we have many excitatory. Are we talking about one excitatory neuron here and multiple inhibitory inputs, or? Yeah,
you can think about one excitatory postsynaptic neurons that it is active at some point, right? So it has a peak of activity at some point. Then in these model networks, what happens is that it receives many connections from inhibitory neurons that are active at different points with respect to this postsynaptic excitatory neuron. And so basically in the model, the mechanism by which the postsynaptic excitatory neuron is shut down before or after its peak is because it receives a lot of inhibition from neurons, from inhibitory neurons that are active at different points. Right?
So you're saying,
so whenever,
yeah, go ahead.
Yeah, so you can imagine excitatory neuron that has a peak, let's say at time 2, then it must receive strong inhibition at time T1 and T3 to be shut down at time T1 and T3.
Okay, and that's so the red curve. That's when the inhibitory neurons are active at different time points relative to the excitatory neuron.
Yeah, so the peak of the excitatory neuron would be at 0.
Yes.
Then,
and then the
far
y-axis
from 0,
and the y-axis
is the current from presynaptic neurons.
So and then on the right side, you're saying that the sequentiality is lower when we get this input from inhibitory neurons at different time points. So
what I'm saying here is that if you reduce the strength of this diff connection, then you get less sequence.
Right.
So having a strong inhibitory current coming from inhibitory neurons that are active at different times is important to keep the sequence.
But reducing it from the ones that are active at the same time as the excitatory neuron does not change things.
Yeah, exactly.
Okay. Right. Okay.
And I mean, I think it's important because in the literature there are different hypotheses about how sequences are generated. So one is, feed-forward chain or syn-fire chain or whatever, where you have excitatory connection from one neuron to another. So this, in principle, doesn't rely on inhibition. This suggests that in this kind of network, at least inhibitory connectivity is important for.
But the sequentiality we get across the excitatory neurons, not just in one. So then is it that like we have, let's say, 2 excitatory neurons, and then each of them gets an inhibitory input that is far from its...
Yes, so this figure here is an average over all postsynaptic.
Right. And then how about the inhibitory population? Do we see that sequential activity?
Yes, there is also sequentiality in inhibitory population. Honestly, I don't remember if this figure is only from an average of excitatory or both excitatory and inhibitory neurons, but my guess is that the mechanism would be similar for inhibitory neurons, but we see sequences also for inhibitory neurons.
Thanks.
This doesn't really give you a mechanism of propagation of the sequence. Just gives you a hint of the mechanism by which neurons have a peak, let's say, but not how it propagates the sequence forward.
Right. Thanks.
Yeah. All right. So just take-home messages are that both E and I cells in the association cortex in a decision-making task are trial-type selective, and we see co-selective E to I and anti-selective I to E, and network models suggest that this circuit motif may be used to enhance the encoding of trial type or choice in the neural activity through this opponent inhibition motif. So just quick follow-up. So connectomics is not the whole story. So, because circuit function dependent on other factors such as external input or the operating point of the network, basically where it is on the transfer function, which can modulate the effective connectivity, or neuromodulators. So a couple of questions that we asked is, how does structural connectivity correlate with functional connectivity, and how does structural connectivity, so anatomical connectivity, correlate with causal connectivity? So what we mean by functional connectivity, so here's just noise correlation, which is the Pearson correlation between single trial activity of a pair of neurons. So at fixed stimulus is the correlation, single trial correlation between neural activity. And what we found is that for E to I connection, the strength of the connection positively correlated with the value of the noise correlation for pairs of neurons. And for inhibitory connectivity, you have the opposite trend. So the connectivity strength is negatively correlated with the noise correlations. So yeah, excitatory inhibitory connections then predict the strength of noise correlation or functional connectivity across pairs of neurons. And for the second question,
Giulio, sorry about the noise correlation. So this is about E to I or I to E connectivity. Which one predicts the strength? Both.
Both, yeah, both, because you would expect a positive correlation between the two for excitatory connectivity and a negative correlation for inhibitory connectivity. So meaning that for a pair of I and E neuron connected by I to E connectivity, you would predict that the stronger this connection is, the more negative the correlation between single trial fluctuation in neural activities. Right. All right. And so for the last question, so how does structural connectivity correlate with causal connectivity? We're currently collaborating with the Harvey lab. So we're doing all optical approaches in PPC, doing the same kind of task. So it's still decision making during navigation. And so here we combine two-photon and single neuron perturbation during behavior. And so what we do is perturb single neurons in PPC in two different epochs of the task, either during the Q period, when the animal sees the visual stimulus, or during the delay period, when the mouse has to remember the stimulus. And so just preliminary results. So what we find is that during the Q, we have, we measure this causal connectivity, which is measured basically by computing the response of non-targeted neurons, so neurons that are not targeted by photostimulation. So the response of non-targeted neurons during stimulation trials minus the response of the same neurons in control trials. So it's basically quantifying the effect of photostimulation of a given neuron on other non-targeted neurons. And so if you plot the causal connectivity strength as a function of the selectivity similarity between the targeted neurons and the non-targeted neurons, you find that perturbing a given target neurons would excite more neurons that are similarly tuned with the target neurons and excite less neurons that are oppositely tuned with respect to the photostimulated neurons. And so this suggests a motif that is consistent with opponent inhibition, meaning that you have more excitation within pools of neurons that prefer the same stimulus than between pairs of neurons that prefer different stimuli. So this is during the Q period, but if you do the same analysis in the delay period, we found something different. So here, the picture that you get is the opposite. So perturbing a given target neurons would excite more neurons that are oppositely tuned to the targeted neurons. And so we're currently working on this. So I think it's interesting that in the Q period, where the mouse has to kind of build an internal representation of the cue or integrating the sensory stimulus, we see something compatible with opponent inhibition, which is consistent with the anatomical data. But in the delay epoch, we see something quite different that we still need to make sense of, actually.
One question about here. This is very interesting, this part. And then in James' paper, the one after me from Anne's lab, James Roach. So if I remember, they were showing that, so for the two architectures, same to same or like same to different, that one optimizes speed and the other optimizes accuracy. So depending on what your on the demands of your behavioral task, we may have these different forms of connectivity between E and I. That inhibitory may connect to same excitatory or to different excitatory. And then this is kind of a line, not necessarily a line, but this reminds me of that here also maybe initially during the queue, because this is more about than accuracy here, and delay is more about speed, because your speed version, sorry, not your speed, your delay is more like the same. So now you see that when the selectivity is high, causal connectivity is negative, which means that the neuron, that the excitatory neuron inhibits the other excitatory neuron.
Yeah, exactly.
So then this will go through the IE different connectivity. And the other one is more I2E same connectivity.
Wait, say that again, sorry.
Okay, I guess let me ask you this instead of me trying to guess. So the I2, the different architectures for I2E connectivity, which one is aligned with, which one goes with which one of these two cases?
Yeah, the one where you have opponent inhibition would be aligned with the Q period, right? Because you excite more the neuron that prefer the same stimulus. And you tend to inhibit or excite less the neurons that prefer opposite stimulus.
And the other one is more same inhibition.
And the other one is, it would suggest more like feedback inhibition within the same pool of neurons.
And then I guess what I'm saying, if I remember James' paper correctly, that they propose that opponent inhibition is more aligned with a decision circuit that prioritizes accuracy over speed, while the other one prioritizes speed. I might be wrong, but I guess that kind of intuitively makes sense. So I'm saying I'm curious about how this Q and delay period could get linked with that idea.
No, I think it's really nice observation. Right now we're working on a different hypothesis, which relates to the decision during the stimulus integration during the Q period and then sequence propagation during delay. But I think, you're right. So I might.
Interesting. I see.
Back in that paper. Yeah.
I see. And then why would sequence propagation require same inhibition?
So we did an analysis where you look at how the vector that contains basically this causal connectivity effect aligns with different dimensions of the of the network, and it seems that what the network does is to basically inhibit the, let's say, population activity at the current point and excite population activity at later time points. So if you would do this analysis on later time points, you will see something more similar to the queue. So right now we're thinking that this may support sequence propagation in the sense that you inhibit the current activity and excite later population activity vectors. But yeah, it's still preliminary.
And one more question. And so if you're proposing here that this I2E connectivity is dynamic, it could be opponent or same, then the data you showed, the EM data, I guess from there you concluded, was it from your EM data that you concluded it's more opponent inhibition?
Mm-hmm.
So then how
Yes.
come, is it because the opponent inhibition is the dominant one, and that's why eventually in these slices we see that? But so I'm saying if it is dynamic, then how come we just found that?
Yeah, yeah, that's a good question. I think we don't have a good answer for that. So I guess there could be a substrate of anatomical connectivity, which is structured in opponent inhibition ways, and that's that corresponds to the effective connectivity during the queue. But then maybe during delay, you have different activity levels, and the network could be in a different state, so that also modulates the strength of the structural circuit. And yeah, that might be a reason why we see dynamic effective connectivity throughout the epochs. But that's something that we need to think more about.
Sure. Thank you. We can proceed.
Yep. All right. So yeah, I just added to these 2 points. So structural connectivity in PPC is consistent with functional connectivity or noise correlations, and this anatomical connectivity in PPC, as I said, is consistent with causal connectivity obtained through optogenetic experiments during the Q period, but not during the delay period. And yeah, so just here for the future direction would be having larger anatomical connectivity data set, especially for E2E and I2I connectivity, for which we didn't see many connection. And then it would be nice to constrain circuit models of decision making using optogenetic data, and as we just said, reconcile observation from anatomical and causal perturbation data sets. Yeah, I just thank again my collaborators, and thank you for the invitation. Super nice. Thank you.
Very, very nice, Giulio. Thank you. Do we have any questions? Go ahead, please, Drew.
Hi there, can you hear me?
Hi, yeah, I can hear.
Great, wonderful talk, Giulio.
Thank you.
I had 2 questions. I'll ask one and then the other. So the first question I had was, you discussed the importance of this, it's kind of like almost contrastive pattern, and that it allows you to accentuate the decoding accuracy. And I'm wondering, might you see some utility also in the opposite pattern, where essentially 2 similar inputs are pushed closer together? So oftentimes when we think about decoding problems or pattern recognition problems, one class of problems is what's called pattern separation, where you have 2 similar inputs, you want to split them further apart. Another class of problems, though, is a pattern completion problem, which is you have 2 different inputs, but you want similar ones to be pushed closer together. And so do you see this kind of motif as being able to, or this framework, I guess you have, as being able to kind of address both of those sides of things? You focus on the pattern separation side, but, for instance, like in hippocampus, you think about an area like CA3, where people think of it as being kind of this auto-associative network that's supposed to push distinct patterns apart, or you think about dentate gyrus being one that's a separator as opposed to really split apart similar input vectors.
Yeah, for sure. I mean, so there is a continuum of the circuit structure where we can go from opponent inhibition to more like within circuit feedback inhibition, and that might, as you said, underlie different computations. So I agree with you that this other extreme may subserve other computation. So in this case here, where you have two choices that needs to be discriminated, I think it makes more sense that the network wants to push apart these choices and do this through opponent inhibition. But I think in other areas or for other computation, you might want to have these other circuit motifs. But I think this depends on the specific areas or specific computation that you want to achieve. But for sure, I mean, this is not to say that this motif is not useful. I think it is, as you said. Yeah.
Great. Okay. So the second question I had was, you identify this motif and it seems really well tuned for allowing these ensembles representing different task conditions, left versus right, being able to be better separable. But I'm wondering, given the density of inhibitory connectivity, and given the fact that PPC isn't just solving this one task in an intact normal organism, it's doing this, for potentially hundreds of possible situations in animals in the environment. Do you think that this kind of circuit motif either can generalize across tasks, or do you think that essentially the population activity for, let's say, left versus right is going to be on some low dimensional space, and in other tasks it's also going to have the same separation of cells? That is, that if I'm two cells that encode left in this task, in a different task, which might have different stimuli and different goals for the animal, those two cells will also be more correlated in terms of what their activity is, and so you don't have to have any adjustment to the inhibitory feedback motif.
Let's see. No, I think it's. Yeah, I'm not sure I have a good answer for this. I think it's totally possible that this motif depends on this specific task that the mouse was trained on, and right now we don't have any mean to say anything more concrete. But I mean, maybe I can answer you with also this example here. So this maybe suggests that also in the same task, the network might want to do different computations that depends on the specific epoch that the mouse is in, right? So what we explore in anatomical data is just one computation, which is the separation of choice. But then, PPC, local PPC circuit might want to do different computation depending on the epoch. So the connectivity might want to restructure, reconfigure during the task, and that seems to be the case. Yeah, maybe the opponent inhibition motif is just a kind of main connectivity background over which different computation might be kind of restructured during the task.
But that can't occur on this time scale of a few seconds. I mean, that's
yeah.
So do you think that, your model is a linear model, if I understand correctly? And do you think that perhaps the only way to rescue that then would be to put in nonlinearities in terms of the transfer function that the neurons have, or some kind of interaction terms in the dendritic integration that could possibly get that? I mean, you can't rescue it because the structure's not going to change fast enough. So I guess it would have to be that the units that you're using in your network need to have more complex integrated properties to resolve that.
Yeah, absolutely. Yeah, that's what I'm thinking about within the same task. So
Or
for the
or that it's still linear, but both of these connectivity patterns exist, but they get activated at different time points.
it could, yes, okay, so yeah, it could be linear, and then you have different circuits in the same linear network that take care of different computation, or the same circuit with the same connectivity that gets reconfigured through nonlinear processes. That's yeah.
So would one way to get roughly that would then be that the ensemble that's doing representing the Q would be different than the ensemble representing the delay, and those different ensembles would then have different connectivity motifs.
Yeah, exactly. But then this it's a bit harder to reconcile with the anatomical data, I guess. Yeah.
Yeah. Yeah. Interesting. Thank you so much.
Yeah. Thank you.
Very cool. Okay. I guess if we don't have any more questions, we can wrap up. Thanks again.
Thank you. Thank you very much.